My name is Euan. I'm a senior research associate at the Natural Resources Research Institute, which is it's an institute at the University of Minnesota Duluth. I've got a list of people on this opening slide here. I want to make sure I'm not pretending to take credit for the work that lots of people have been doing in my group for many years now. This is the current team. The list would be massive if I actually had everyone in there. But this is I'm going to present some old results from the Great Lakes and some new things, some kind of confounding things that we're finding in the Laurentian Great Lakes that are not fully explained yet. Should I just get going? Is that the idea? Just jump right into it?
Yes, that would be great. Thank you.
Right. Okay, so first of all, let's talk about the Great Lakes again. If you work on like Baikal or Titicaca or whatever other lakes, it's not a competition. I just want to just illustrate the importance of this water body to the continent of North America. I mean, it is the largest surface freshwater resource in the world, combined. And if you live within the catchment of the Great Lakes, you probably think about it. Outside the catchment, I'm a little surprised sometimes of the poor understanding of the Great Lakes and their importance on the continent. I think as decades pass, long after I'm dead, their importance is going to increase more and more. I think that motivates us to study it, not just to sort out the problems that the Great Lakes have right now, but also, predict where things are going in the future for this resource. There are other states well outside the catchment that are thinking about how they're going to tap into this Great Lakes resource in the future. And so these are things that we like to think about whenever we're making predictions for the future. And so there are all these great statistics about, the size of it and the number of people who rely on the Great Lakes and so on. My group has been really fortunate to be part of this Great Lakes biological monitoring program, which is in partnership with the US EPA out of Chicago. This is a program that's been now running for 40 years, where all of the Laurentian Great Lakes, and particularly the deeper waters of the Great Lakes, are sampled for 2 or more months out of the year. Folks in my group spend a lot of time out on this ship, the Lake Guardian, collecting samples. Our focus is on the phytoplankton side of things, which we've expanded quite a bit over the years. We work with a larger team of scientists who are sampling water quality, zooplankton, benthic invertebrates. We've introduced a paleo program starting in the earlier 2010s, which I'll mention that we are continuing to expand in the Great Lakes system to look at history. This was initially established under the Great Lake Water Quality Agreement, which is a subsidiary of the Clean Water Act, largely in response to the craziness of Lake Erie in the late 1960s and early 1970s. To monitor the system, start tracking what's going on, and hopefully identifying problems before they become huge problems, as happened in Lake Erie. This is what the network looks like. Okay, so the primary network that has been sampled for the longest on the map here are the little red squares. Those are the deep waters that have been sampled in many cases since 1983. I want to just mention that the bluish circles are locations in the near shore. As we started to characterize the deeper waters and start to get a good handle on it, we started pushing EPA to move into the near shore areas where we're starting to recognize a lot of localized problems taking place. And so the blue points are ones that we've now added to the system for regular sampling. The yellows, in particular, the yellows represent places where we have cored or we are going to collect sediment cores in the future to get a representation of the long-term history of the system. A number of you online, I'm sure, are familiar with paleolimnology. We use it in the Great Lakes, mostly in an Anthropocene context, maybe going back 300 years to look at the long-term anthropogenic history of the system so that we can, see if things are getting better, where remedial action plans are in place? What are the causes for a lot of the changes in the system? And hopefully in the future to use these paleo data as an early warning system. I'll talk about that a bit more in a minute. This is the primary device we use for sampling phytoplankton, the rosette sampler that is lowered off the Lake Guardian. Again, we are going back to the same locations every year in the spring and the summer. Just some basic data. These are data that we need to update our publications on this, but some of the major trends that we have observed through the collection of phytoplankton out in the Great Lakes. For instance, the fairly recent invasion of the quagga mussel has obliterated the pelagic food webs in Lakes Michigan and Lake Huron. Okay, so these are huge water masses that have lost incredible amounts of diatoms out in the open water as these filter feeders came in. And these are not like zebra mussels, which are basically limited to nearshore shallow areas. The quagga mussels have a form that has adapted to the deeper waters. So the entire basins of Lake Michigan and Huron are coated with these things. So anything that is sinky tends to be at a disadvantage. And that includes the diatoms that are have been substantially reduced in these systems since they were introduced. Other things going on, the opposite kind of things in Lake Erie. Again, something we thought we dealt with in Lake Erie was the remediation of eutrophication following the installation of the Clean Water Act. But then if we start looking at recent data, it's more than just anecdotal observations. We know that things are getting worse in Lake Erie, and there are new reasons for it compared to the 1960s. And you can see quite clearly in these long-term data, the increase in the cyanobacteria relative to other species in the Lake Erie system. And so I just want to mention quickly, again, nothing super new here, but we've been collecting sediment cores throughout the Great Lakes system. Just here's a picture of coring in Lake Superior. These are data that have led to a number of papers, a number of separate studies. We have basically published on the paleolimnology of the deep waters of all the Great Lakes so far based on these paleo data. And often we bring the data from the entire Great Lakes Basin together for all the lakes, despite the fact that every lake has its own concerns in terms of environmental stressors. We published some results on the long-term changes in the lakes. I've been modifying our approach to this to try and support remediation and future monitoring plans by using paleolimnology as an early warning system, kind of like what we have in our weather systems. We have early warning systems because we have all kinds of great satellite data that tells us when storms are coming. Why not use the fossil record as well as an indication of changes that are taking place? We can look at hundreds of years, but then focusing on the most recent 30, 40 years and look at trajectories based on the fossil remains, not just diatoms, but also algal pigments, geochemistry, all these other things you find in the fossil record to tell us about the long-term history. This has really come through in our evaluation of the cyclotelloid diatoms in the Great Lakes, where no matter where you are in the Great Lakes, these cyclotelloids are what you might want to call Cyclotellus sensulatu, or a number of other species. For instance, this is Pandixiella laurentiana, a new species that we described back in 2018. These are increasing in a relative proportion in all of the Great Lakes based on the fossil record. So we know the Great Lakes are getting warmer. Our initial hypothesis was, well, what's going on here? What could be happening across all of the Great Lakes simultaneously? Lake Erie is getting more eutrophic. Lake Superior is ultra oligotrophic. With all these other unique drivers in each Great Lake, why are we seeing something that is changing consistently across the entire Great Lakes Basin? And so our immediate hypothesis is, well, the atmosphere is warming, and this is satellite data on the surface water temperatures across the Great Lakes, and there is a significant increase in these surface temperatures in the last 20, 30 years. undoubtedly changing the physics of these systems, and I would imagine having some impact on the primary producers out in the deeper waters. And so what we saw is no matter where you are in the Great Lakes, there is this relative increase in the proportion of these cyclotello diatoms. Again, there's that Pandixiella laurentiana, delicatula is another very common major diatom player in the Great Lakes. And they have been increasing in proportion in the system. And so and I'll kind of show a slide like this a few times here. I mean, who really cares that we have nothing really to fear from these centered diatoms, but they do reflect a change in the system across the entire basin, which no doubt is represented in greater parts of the base of the food web than we realize. And so I'm going to come back to that. So that was one of our first sort of early warnings that something is changing there. We see it changes rapidly taking place in the diatoms. Undoubtedly, it's happening in the zooplankton. We just don't have the sampling resolution to see it, or we just haven't done the paleo studies to see it yet. And like I mentioned, we've been moving into the near shore in the Great Lakes. We have all kinds of problems going on locally. Every port around the Great Lakes has its own issues associated with their own stressors. It could be climate, but also things like invasive species, changes in water levels, agriculture, all of these stressors that are unique based on where you are around the catchment of the Great Lakes. And so we have now moved into studying the near shore, not just the deep waters of the Great Lakes. In particular, I wanted to mention back in the early 2010s, we had a goal of determining whether the goals of the remedial action plan for the St. Louis River estuary in Western Lake Superior have been achieved. Ever since the St. Louis River estuary was established as an area of concern in 1987 by the EPA, we now have state-of-the-art sewage treatment. And so we aim to use paleo data to evaluate in particular this beneficial use impairment of excessive loading and nutrients and sediments in the system. So that involved a suite of sediment cores throughout Western Lake Superior and into the estuary and the harbor. Collected some sediment cores. This is back in 2014. Piston coring, again, we're looking at the Anthropocene. In this case, we had about 250 years worth of sediment, which in the estuary ranges from about 50 to 100 centimeters of sediment accumulation in the system. We have our established diatom-based models to reconstruct phosphorus concentrations in the system and use this to perform reconstructions. And indeed, since the awareness of this problem, the installation of sewage treatment, known improvement in the effluent to the system, we see things got better in Western Lake Superior, as indicated by the diatom assemblages. Okay, so we saw this early 20th century rise and then a more recent fall in the inferred phosphorus in the system. Again, it's always been an oligotrophic system, but it underwent some cultural eutrophication temporarily and then came back from that brink in more recent times. So indeed, things got better, and we were able to use the paleo data to confirm that. But now, combined with a lot of anecdotal and very real scientific observation that algal blooms are becoming a greater problem in Western Lake Superior, there are arguments out there that the algal bloom issue is due to greater anecdotal and scientific observation. More people are thinking about it. There are more observations now. When it comes to Western Lake Superior, it does appear that it's becoming a much greater problem. Even in these oligotrophic waters along the south shore of Lake Superior, we're seeing algal blooms even out by the Apostle Islands in Lake Superior. Okay, so not necessarily in protected bays. This is a photo taken recently behind Barker's Island, which is in the harbor in the St. Louis River estuary of Clique phanezomenon, in blooms we've been seeing recently, and very recently have been detecting actual toxic components as a result of these harmful algal blooms in the system. So there have been greater issues recognized in Western Lake Superior and in the estuary. And so going back to the paleo data, we can see that based on the diatoms we saw increasing in the system, it's this grab bag of all the diatoms you don't want to see increasing. I mean, again, we used a diatom-based reconstructive model to give quantitative information about nutrients in the system, but I just want to give a picture that we saw all of these changes in recent times, a very rapid increase in recent decades of all of these high nutrient indicators in the system, for instance, in this little bay called Aloise Bay within the harbor. And then using the reconstructions, we were able to see that when it came to these more protected bays and nearshore areas, things appear to be getting worse in confirmation with all of these observations of the blooms in the system. So increases in phosphorus levels in the system, and also, I'm not going to show all of the data here, but also increases in cyanopigments in the upper portions of these cores, all suggesting that this is an increasing issue in the system. All right, so I'm going to come back to that. I want to talk just a little bit about some of the other changes we're seeing in the Great Lakes. I want to establish to everyone that there's a lot just changing in the Great Lakes. It's really hard to keep up with everything that keeps shifting in the Great Lakes and trying to come up with reasoning for it all. And now that we have been involved for, we now have about 20 good years of detailed phytoplankton data from the Great Lakes. We have the ability to look at some of the details of the changes at a species level that are taking place within the system as well. So our group is responsible for this so-called State of the Great Lakes report, which comes out every few years to make interpretations of where things are good and bad in the Great Lakes system. And so we've got phytoplankton at the top of the list here. I'm responsible for writing that portion of the report as a sub-indicator. And we have these interpretations, which are sort of based on the very basics of the interpretation. Although I keep saying that things are changing in the system, when it comes to the things that people care about the most, you know, when we say Lake Superior, we say it's unchanging because it's not necessarily moving to a place that's bad. We don't fully understand it yet, but it's still a very oligotrophic system. We see deterioration in Michigan and Huron because of the invasive species issues. Lake Erie, like I mentioned, is getting bad, and so on. And so I'm responsible for reporting back on this for the Great Lakes as a whole. And again, looking at Lake Erie, the monitoring data, these are the sorts of data that the majority of folks and the funding agencies care about seeing. Okay, this is the first thing they want to see is what's going on with these major algal groups. Again, cyanophytes increasing in Lake Erie. But then when we start looking at the species, we can see that there are specific cyanobacterial species that are increasing. So whenever you see these plots, I'm going to throw a lot of data at you here, but whenever you see these plots, these are just the abundance data for the individual species. If you see a regression line, that means there is a significant change over the time period represented based on a non-parametric Kendall test. Okay, so we can see, for instance, there is this increase in Aphanotheke in Lake Superior, as indicated by the blue line. Lake Erie showing an increase in Aphanozomenon, and so on. So again, we can look and see what the actual species are now, which is going to gradually help us get at what the drivers are of the changes in the system. Again, more of the basic data. We saw this dramatic decline in the abundance of diatoms in Huron and Michigan following the invasion of the quagga mussel. And whenever we look at the data for years, we've been saying, okay, well, this change is because of the loss of diatoms. Some of the major players in terms of biovolume are Fragillaria crotonensis, Stephanodiscus subtransylvanicus. Again, you see these lines, significant declines over this approximately 20-year period from the Great Lakes based on these monitoring data. And we've been saying for ages, well, it's probably because the quagga mussels, these filter feeders are coming in. You're going to lose the heaviest algal organisms in the system. That's the most obvious change. But then you see things like a blue line for Lake Superior. Okay, so that the quagga mussel has not substantially invaded Lake Superior. Why are we seeing declines in the abundance of diatoms in a lake that is not loaded up with filter feeders? Okay, so this is now for us moving beyond just invasive species in the system. And so here are those long-term paleo data on the cyclotelloids that I showed before. But then whenever we look at the individual species, again, Pancasechiella delicatula, this diatom that is a major player throughout the Great Lakes in the summertime and into the winter, we see, despite the fact that it's increasing in relative proportion in these sediment corridors, it's declining in abundance over the years based on the monitoring data. At first, that didn't make a whole lot of sense to me. And then we've come up with a working hypothesis that what may be happening here is because we have basically have warmer lakes. If we have an extended summer season that's moving more into the fall and possibly even into the winter when these cyclotelloid diatoms are thriving, we are just getting a longer period of these cyclotelloids being fed into the fossil record, despite the fact that they appear to be decreasing in our spot synoptic sampling over time. So that's a working hypothesis at this point. I don't think it's necessarily a conflict in the data. All right, so let's move into, again, I keep showing these title slides because this is several talks crammed into one that have been presented at conferences and so on. And so looking at some of these data now, again, these abundance plots, the upper plots, again, are the ones that the agencies are most concerned with. They want to know what amounts of algae, how much food is out there. We, of course, are interested in getting into the nitty-gritty of these data. And for instance, we started to notice the proportions of certain organisms were increasing. You know, these lower plots, we sort of made them small because in general, most people didn't care about them. But we started to care whenever I started to notice that it looks like the proportions of certain organisms in the case of Lake Michigan, cryptophytes in the springtime, and Lake Ontario, its dinoflagellates are increasing in relative proportion. Again, a ton of data here, but then whenever we started to look at these individual taxa of flagellated organisms, we see this resounding increase in flagellates throughout the system. And again, when we first noticed this, we're like, oh, well, again, maybe it's invasive species. Maybe it's the quagga mussels coming in, and these things that don't so easily sink have an advantage of keeping themselves up in the upper water column. But again, we see this rise in Lake Superior where these quagga mussels have not heavily invaded the system. So there's more going on here than just loss of diatoms because of mussels feeding on them and flagellates coming in and replacing them. So again, who cares? Well, first of all, just talking about the cyclotelloid issue, this is a slide I made a few years ago when we published that work, that we're seeing a clear shift of these large filamentous diatoms like Allosyra to these smaller single-celled cyclotelloids out in the open water. But I now expand that to the loss of diatoms and the shift to the flagellated organisms in the system. There has to be a greater ecological impact due to this. You know, the zooplankton have feeding strategies. There's got to be something else important going on here that we don't fully understand yet as this change is taking place. It does not appear to be a top-down control. It may be related to climate change and some other things taking place, but for now, we mostly know that a change is taking place, and it is up to us to now sort out what is it that's driving this change. If we have a hope of managing it, if it's something we should actually be concerned about, we need to sort out what is causing the change. And so just this is an example of a plot I made for our Western Lake Superior, our St. Louis River estuary study, representing the types of shifting in the stoichiometry in the system that may result in, for instance, harmful algal blooms that we're seeing in Western Lake Superior. In this case, this would be, you know, this is a mock-up of a hypothesis that a storm drives the changes in the different nutrients over time, resulting in cyanophyte blooms, changes, loss of the diatoms, and so on. This is just hypothesis at this point for the near shore, and this is something that we need to expand into the offshore as well. So looking at the deeper waters of the Great Lakes. On the left, here's some summary of the spring single-celled flagellates, the summer colonial flagellates in the system. Again, dominant rise over time. And there's no analysis here. These are just plots of data. We see that all kinds of things in the Great Lakes are simultaneously changing. The ratio of nitrogen to silica, which has implications on diatom assemblages. Again, when you see a black line, that represents all of the Great Lakes data combined and the significance of that change taking place, as well as the color-coded individual lake analyses. Beam attenuation, that is basically telling us that the water is getting clearer, all right? And chloride, you know, these top two, are they cause or they effect of this? You know, it might be a bit of both. Chloride, we know for years, has been increasing in the system. Is there something with chloride or what is associated with chloride that is driving this change? Because we know wherever you are in the Great Lakes system, chloride is going up, which appears to be a legacy road salt effect. Whether that's driving the phytoplankton, we don't know. But we are starting to analyze these relationships between water quality and the different types of algae, sometimes down to the species level. This is very piecemeal at this point. For instance, this is just showing total phosphorus on the left, the nitrogen to silicon relationship in Lake Michigan related to the abundance of flagellates in the system. Okay, so we see these significant relationships. And I show this, I say it's piecemeal because we're kind of, you always want to have lots of data. And the problem with having lots of data is that it just confuses everything. We're trying to bring this together and identify what the drivers are of this change. And because we have so much data and so many variables, trying to crunch that into something that's informative has been a real challenge. You look at these independent analyses like this, these piecemeal analyses, and they may look good. You might say, okay, maybe there's some relationship here, but there's so much intercorrelation among all of these variables. So that's something that we are gradually sorting through. Talking about the nearshore environment, one way we are doing that is we need to establish monitoring programs. We have a great Great Lakes deep water monitoring program established. We want to also start establishing locally, and we are currently focusing on one in Western Lake Superior in the estuary at Duluth Superior. These are locations that we are intensively sampling right now for a full suite of water quality data and algae as well. Lots of time at the microscope. We are collecting sample sets sometimes throughout the summer as we get close to harmful algal bloom periods, sampling once a week, sometimes even more frequently, collecting the actual algae and the associated water quality data. So a sample set includes actual microscopic assessment of the phytoplankton in the system, as well as a whole suite of the potential drivers of the algae in the system as well. Okay, so this is something give us another year and we'll start reporting on this. But the idea is we need to bring these data together to identify, first of all, do we have enough information to characterize the system? That's some type of redundancy analysis. How frequently do you need to be sampling? What parameters do you need to be sampling? Where do you need to be sampling? All of these things we need to sort out if we have a hope of coming up with a useful monitoring program for Western Lake Superior. And of course, this evaluation of cause and effect that I've been getting at is extremely challenging because you are taking highly complex data sets and trying to crunch them down into something useful. Another confounding effect is the fact that, you know, we're not dealing with monocultures here. And this is a paper we recently published. I was involved in this because I recognized full well, even during these algal bloom periods, there is such a focus on the cyanobacteria. But we know from looking at these samples that there's much more in these samples than just cyanophytes. You've got a mixture of diatoms. Sometimes it seems like the same diatom species are hanging out with the cyanobacterial blooms. You know, whenever you do correlations between species, in this case, you know, one diatom versus two other diatoms, we're not surprised when we see significant correlations between two cyclotelloids. They both hang out in the late summer, probably have similar chemical dependencies. But then we see other things like the cyanobacteria. In Lake Erie, one thing I've noticed is that highly abundant Synechococcus, a cyanobacterium, always seems to be hanging out with Algae granulata. Why is that? Is it do they just like the same things, or are they hanging out together for some functional reason? And so, getting into some of the more of the technologies of these analyses, we are attempting a clique analysis. So the idea here is if you can come up with cliques of taxa, groups of taxa that tend to hang out together, maybe we have a hope of actually analyzing against potential driving variables and explaining what's going on. Again, I'm trying to get at this collapsing of complex data sets. Just doing another NMDS or a CCA is fine for looking at the data, but we need approaches here that go beyond ordination and ANOVAs to try and identify what the driving factors behind changes in the phytoplankton in the system. This is an example of another paper by Fullerton in Nature Geoscience. These are DNA data that identified hundreds of different bacterial species in a deep water marine rift. But we took this and said, okay, well, let's try the same principles in our Great Lakes species identified via microscopy. And what we found is that there appear to be, at least based on our spring and summer collections, there appears to be 5 distinct cliques of phytoplankton in the system, which we can summarize like this. Again, a lot of information here. This is something we're sorting out, but it's pretty clear. There's a clique of the summer bloom. Alocosira islandica, the most common diatom, or most common alga overall in the Laurentian Great Lakes, has a clique of associates. And then there is a group that is not associated with Alocosira. We can take this information and maybe actually relate clique abundances with the long-term data. Indeed, we see good relationships whenever we take, for instance, our clique number 2, friends of Alocosira, related to total phosphorus. There's a strong relationship versus our clique number 4. There's a clear decline. And this is maybe an approach that will help us get at what the drivers are instead of trying to do things piecemeal, species by species, or across all this whole suite of water quality data. This might be something that can help us out. And again, this is the last time I'll show this slide. Who cares? Maybe nobody. We'll see how it goes. This is something that we're attempting now, but again, I mentioned we've tried this clique analysis among others to try and get at the complexity of these data, which is difficult to work with whenever we are trying to come up with real management recommendations from these results. All right, so let me just wrap it up. If there are take-home messages here, the largest surface freshwater resource in the world has a food web base that is reorganizing, not just changing in abundance. Okay, the abundance changes we've known for years, but there's a lot going on within that. Even if abundance, say in Lake Ontario, is not changing, the composition is definitely changing at the base of the food web. And so there undoubtedly, if this is going to continue, there are going to be implications on food webs. It's eventually going to get up to the things people care about, like the fish. And so we're working on the drivers, such as, maybe using new techniques like clique analysis to get at why these changes are taking place. I just want to also mention quickly, the latest addition to our program is a suite of paleolimnology assessments that we have just started getting into. In case anyone is interested where we're going on this, this is our proposed list. Some of these have been sampled. Many of these we are going back to revisit. Locations visited by Gene Stormer, who focused on some of the very early diatom studies in the Laurentian Great Lakes. So we're hoping to give updates on the paleolimnology of that system. Again, moving into the near shore. So I feel like we've done some pretty good due diligence in the deeper waters. Let's move it into the shallower waters where there are localized impacts, or maybe there are overall climate effects. Let's find out. Let's leave it at that. Thanks.
Hey, Euan, great, thank you. And also one of my takeaways there. I love that you have paleolimnology as an early warning tool. I've never thought about characterizing like that. So that was really great. I have a lot of questions. We have the luxury of being able to chat with those at other time. So I'll go to Lawrence has a question in the chat. If anybody wants to ask Euan a question, please raise your hand or drop it in the chat, and we'll get it to you, to Euan. So Lawrence asks, and I think you were just getting at this, but what do you think the implications of the shift from diatoms to flagellated groups might be?
The implications on the lake condition, or
I'm maybe guessing the food web. Lawrence, if you want to expand on that. I didn't know if maybe there's maybe a more direct impact to the zooplankton having a more mobile alga becoming dominant. Yeah, Lawrence was asking about the food web.
Okay. So I mean, yeah, these, a lot of these like cryptophytes, things like that, these tend to be small, tasty algae that are preferred by a number of the smaller zooplankton. We are seeing all kinds of what appear to be changes in the zooplankton that we had associated previously with invasive species, but may be related to this change in their food resource. Right now, we don't really know. There are, I mean, even these large colonies of diatoms, a number of the zooplankton would happily feed on them. They're able to chew them up. The diatoms tend to be the best food resource, but a number of species can feed on these flagellated organisms and can't feed on the diatom. So there's undoubtedly going to be some sort of shift in the zooplankton. We just don't have those data yet.
Great, thanks, Euan. Kyle asks, have you examined the correlation of increasing mixotrophic flagellates with fluctuations in bacterial abundance?
No, we just don't have the bacterial data, and we have the way to do that would probably be to start seriously collecting eDNA from throughout the Great Lakes, which is something that we have done. We have introduced molecular collections throughout the Great Lakes. These molecular approaches are not ready for prime time, I have to say. We've had all kinds of difficulties just getting results out of that, but getting the details of the bacteria that are out there, I think that's a big deal in terms of determining drivers. So I don't have an answer for that, but there's probably something going on there. We are just now, trying to characterize the importance of picoplankton out there, or autotrophic picoplankton, and we're having some success there. I don't know if we're going to settle these molecular approaches in my lifetime, but we are trying.
Great, thank you. Miriam says, thank you for a wonderful talk. And she asks, do you ever worry that perhaps some diatomists might count the same data set differently? Is there a way to account for the possible difference in taxonomists or prevent the differences beforehand?
That is an awesome question. And yes, I worry all the time. We've seen it happen. So we have done quite a bit of recounting of samples that were counted by other contractors, and not because the contractors did a bad job. They had their own taxonomic system. When we first got involved in the Great Lakes program, we took on a whole lot of data from the 1990s and started comparing it to newer data that were collected by different contractors, and we could not get them to converge. The species lists look the same, but there are just subtle differences among groups that make phytoplankton data counted by different contractors very difficult to analyze together. The solution to that is, if there's a transition in a monitoring program, that you have substantial training in between. And this is something you've got to pay for. You can't just pass on a program like this to another lab to pick up and start analyzing with their own analysts. You're just going to have problems. You need major workshops. You need major side-by-side analyses. I think it's solvable, but When we first got involved, the EPA really underestimated the effort involved with preventing those sorts of problems.
Great, thank you. Again, Ryan commends you on a wonderful presentation, and wants to know regarding the spring. Oh, my chat just moved. Regarding the spring seasonal diatom assemblage, have you noticed any trends specifically in cold water taxa over time and replacement? The flagellates were mentioned, but are there any early season surprises?
The major early season surprise is that in so many places, with the exception of Lake Erie, you know, the deep waters are becoming like distilled water in the springtime. I mean, that's when we're losing. I mean, that the spring is that's the time for diatoms, and we're losing the diatoms in a big way. And we've had to even change our program. We now collect a full 2 liters of water to process instead of 1, because there's so little out there in the springtime. Part of that is quagga mussel, but again, Lake Superior the same, Lake Ontario the same, get much more productive later in the year. But I would say the major finding there is the loss in overall abundance in the springtime, which is a real thing.
Julianne asks, what do you think is driving deeper light penetration in Superior without the coincidence of a quagga infestation?
Yeah, the only hypothesis I have on that is the physical change in the system due to warming. And we know that Lake Superior is warming faster than possibly any other lake in the world. And that means longer, stronger stratification, and therefore a long-term observation of clarification in the system, because whenever you have a strong stratification that lasts longer, more is sinking out. You still have a few of these flagellates up there, but you're losing solids, you're losing diatoms. That seems to be the best explanation for that.
That's a really interesting perspective on stratification. That makes sense. I haven't thought about that.
I don't know what else it could be.
Yes. Yeah, no, great. Thank you, Euan. Yeah. Anna would like to know, how are you looking at picoplankton?
Oh.
Are you using eDNA?
No, David could even answer that. He's the one actually doing it. So yeah, so we have our standard chlorophyll A protocol out there, which is filtration and analysis using a fluorometer. And we've added to that additional filtering stages. So we have our basic chlorophyll analysis, which uses a 0.7 micron filter. We've now added an additional stage which uses a what is it, a 0.02 micron filter, or 0.2? I'm already
a 0.2, I believe. Yeah.
Sorry, yes, a 0.2 and a 2 micron filter. And so the picoplankton is everything that should make it through that 2 micron filter, and then presumably the 0.2 is capturing everything else, and that should represent our picoplankton. And so that's the approach that we're taking to it. And it seems to be working so far. We haven't done all of the lakes yet, but that's the approach we're taking on it.
Yeah, and Euan, to follow up that on that, you know, we're looking at growth in the micro versus picoplankton and comparing those kind of compartments of the autotrophs. Do we have a step forward or a path forward to do community analysis on the picoplankton? I don't know. It seems like it would drive somebody crazy. But
that's where we need DNA, right? So
yeah.
We certainly don't have the microscope to do it.
So maybe something to look forward to in the future, once we recognize these differences, might be characterizing who is making up those differences.
Yeah, I mean, again, I worry about it a bit. You say, okay, well, you run these filtrations and then do a molecular analysis on the different filters, but you know something is getting through. Can you actually do that and get good data? I don't know. I worry about that. It's not really something I want to take on, but hopefully someone will.
Sure. Sylvia would like to know in your clique analysis, did you find cliques are consistent across the Great Lakes, or are there sub-cliques within lakes?
You could find subcliques. I think we're limited by our taxon list to confirm whether there are subcliques. Those 5 that I showed are the major ones. The original paper, you know, which had hundreds of bacterial species from the deep waters of the marine system. If you had a data set like that, you could probably find all kinds of subcliques based on how you deal with the resolution of the data. In our case, I don't know. I don't have a clear answer for that. We could probably pull out subcliques, whether they're statistically significant, maybe not.
Oh, here's another question from Julianne. Any evidence that buoyancy changes, that there are buoyancy changes in the warmer waters?
Like for cyanobacteria, maybe, is that what we're thinking about? I mean, we certainly, the cyanobacteria have the advantage of being buoyant, many of them. Cyano or diatoms. I don't know. Did diatoms change their buoyancy in response to things like warmth or stratification? I don't know.
Yeah. You and I wanted to ask if you know, something like the culture facilities you have at the ballast water site, might that be an appropriate place to study competition between the small centrics and the cryptophytes and dinoflagellates? I'm just thinking about getting at competition experiments, and trying to figure out why maybe the cryptophytes are succeeding.
Maybe if you can get things to culture in the first place, that's the place to start. And
sure.
So maybe.
Fair enough. Well, great. I'm not seeing any more questions roll in. I could pick Euan's brain all day if everybody wants to sit around and listen to that. Otherwise, I can let folks go early. Euan, really appreciate you coming back and giving another Diatom Web Academy. It's great to have you on. And even though I'm immersed in this stuff every day, it's wonderful to hear your perspectives and kind of the aggregation of a lot of this knowledge. So thank you very much, Euan.
Thanks for coming out, everybody.